This video will provide in-depth technical exploration of XRNA's various supported file formats. XRNA supports a variety of input file formats and output file formats. Number 1. The XRNA file format. XRNA files are XML-like files. They contain hierarchies of elements. At the top is the complex document element. This is the overall parent element. Within the complex document, multiple complexes may be embedded. These are synonymous with RNA complexes. Likewise, complexes may contain multiple RNA molecule elements. Each RNA molecule element contains a single nucleus data element. These are line-separated lists of all nucleotides within the RNA molecule. Each line contains the nucleotide symbol, the X-coordinate of the nucleotide, and the Y-coordinate of the nucleotide separated by spaces. Now let's jump to the next portion of the file. Once the nuc list data has ended, nuc elements begin. These provide the opportunity to change the colors, fonts, and font sizes of this molecule's nucleotides. XRNA is a legacy file version. It was implemented to provide backward compatibility with old versions of XRNA. Therefore, it has some quirks, including some hard-coded features like font ID elements. The next relevant element is the label list element, which may contain a line element, a string element, or both. Line elements contain XY coordinates of label lines relative to the nucleotide which they label. They also contain the stroke width and hexadecimal encoded line colors. String elements contain XY coordinates of label text relative to the nucleotide which they label, a font size element, a font ID element, a hexadecimal encoded string color, and the label content. The final relevant element is the base pairs element. These elements describe helices in a compact fashion. Nuc ID provides the first nucleotide index involved in a base pair. BP nuc ID provides the second nucleotide index involved in a base pair. Length describes the length of the helix. If base pairing between RNA molecules is required, the property BP name can provide the name of the other involved RNA molecule. Number 2. The JSON file format. This is a more modern file format, which is directly compatible with computer languages like Python, JavaScript, and many more. For these reasons, we recommend use of this file format. This file also makes many properties more explicit in contrast with the XRNA file format. You can easily edit properties of a JSON file to make direct edits. Please note that this file is minimized, so not every line is shown. We're going to highlight just a few example elements, so you can understand these files. JSON files are also hierarchical. Parent objects may contain multiple child elements. Here at the top of the file, you will find the classes object, which houses CSS-like style objects, so that they may be reused within this file. RNA complexes may contain multiple RNA molecules. RNA molecules contain sequence elements, which provide the literal sequence of nucleotides within the RNA molecules. RNA molecule elements also include separate objects for labels and for base pairs. Please note that base pairs may be listed in two different locations: within the RNA complex and within RNA molecules. Base pairs listed under RNA complexes may span two RNA molecules. Number 3. The SVG file format. SVG files are image files. They are mostly used for publications and illustrations. They can also be used as input files. SVG files produced by XRNA React include additional information to explicitly describe elements. In contrast, SVG files which weren't produced by XRNA React should be used with caution. We cannot guarantee that they will be imported into XRNA fully or correctly. This is due to the implicit nature of base pairs depicted by images,